to move on to Melissa Omand. So she did her PhD at Scripps and then moved over to Woods Hole to do a postdoc there. And since 2015, Melissa is an associate professor at Rhode Island. I've heard a lot about her work, especially through Minions. So I hope we hear something about Minions today, but if not, it sounds really fun. I like diel vertical migration. So please go ahead.
Okay, yeah, I'll have to come back and talk about Minions another time, because today is, I'm going to present some other work that came out of the exports program, and my co-authors are Deb Steinberg and Karen Stamieskin at VIMS. I'm going to be talking about the cloud-driven variations in the deep scattering layer that arise from variability in the light at these deep depths in the twilight zone. So this is a pretty simple analysis, but the results are pretty exciting, and my talk won't go long, maybe 20 minutes. So first I'm going to describe how we model the light intensity via PAR in the twilight zone down to depths of about 300 or 400 meters. Then I'll talk about how we detected a deep scattering layer from the acoustic backscattering from the 150 kilohertz ADCP. And then I'll compare the layer depths of this deep scattering layer with our modeled isolooms. And finally, look at the swimming behavior, modeling the swimming behavior that we can see based on this correlation, and give some time for discussion. So this work was published in PNAS about a month ago, and we had there are some beautiful pictures from space and from aircraft of cloud shadows cast on the sea surface that, and so we managed to appear on the cover. So the two main sensors, or three main sort of platforms that I'll be describing. The first is just the ship, the surface PAR sensor. So that's an important tool here. The 150 kilohertz ADCP narrow band. This was resolved or processed into 8-meter vertical bins, and it was sampled at 2-minute intervals, which is worth pointing out because the Ravel is, I understand from Jules Hammond, is the only UNOLS vessel right now that actually processes the ADCP to 2 minutes. Standard is 5 minutes. With 5 minutes, we wouldn't have been able to see some of the really cool stuff that we saw. The max depth for the 150 kilohertz was about 500 meters. And so I put this figure from Andoni Lavery at the bottom here to give you a sense of what maybe some of the types of animals that we might be detecting. The bottom line, or the point of this, is that the 150 kilohertz is around 10 to the 5. I don't see my cursor here, but it's in the range of the zooplankton. So our deep scattering layer is likely zooplankton and not larger animals, larger fish or squid. So the third platform that I used is a wave-powered profiling wire walker. This was making profiles down to 500 meters about every 40 minutes. And among other sensors, it was equipped with a miniature PAR sensor. So we were able to get some high frequency variability of the underwater light field. These observations were made from near Ocean Station Papa during the exports cruise from 2018. So this was a 25-day cruise in August and September. What's shown on the right side is ship tracks in gray from the Ravel only, and then red shows the wire walker tracks. And just to put this in context, there was another ship, there was a variety of other autonomous assets out at the time, but these are the tracks that I'm going to be showing from this study. And the takeaway from this, I think, is just that this was a fairly concentrated experiment. So we made lots and lots of measurements in a small space and following the water as much as we could. So in a Lagrangian frame, and that became really essential, I believe, to this analysis. Here's an example of 5 days of the ship PAR on the top panel. and then the wire walker profiles of PAR contoured below. So here we can see some of the variability over the course of the day in the ship PAR that I believe was associated with variability in clouds overhead, and kind of remarkable too, because on the ship, it really felt like it was just drizzly and gray every single day. It wasn't like this was, you know, big cumulus clouds passing overhead and really varying what we experienced as the intensity of the light. But it was still there, and it was enough that you'll see later the animals hundreds of meters below the surface were also detecting these variations as well. So the wire walker PAR has a sensitivity down to about 1 micro-Einstein per meter squared per second. So it really wasn't measuring any detectable PAR below about 60 or 70 meters or so. So that's where the gray transitions to white. So we are really only able to make direct observations of PAR in the upper 100 meters or so. This gave us an estimate of the KD just within the euphotic zone. So the euphotic zone was similarly around 70 meters. The gray dots show the data from the wire walker. And from a linear fit to that, we got an attenuation coefficient of 0.065 per meter. And then below that, I had to model the PAR, and the water below where there's a high density of phytoplankton in the surface is clearer. And so I, anticipated needing to use a KD that is a smaller value than is in the surface. I determined the KD actually iteratively, just by, you'll see later, just by matching the scale of the variations of the deep scattering layer to the variations of the isolume. So it's a little bit circular, but I'm convinced that this gives us a very good estimate of what the KD was. And so now on the third panel, the bottom panel there, you see the modeled PAR between 100 meters down to 350 meters. And so, because we're using this constant KD in two parts, in the upper and the deeper water column, all of the variability in the surface PAR is now translated to isolumes hundreds of meters below the surface. And so we have variations in our isolumes that reflect some of the variations at the surface. And now we can compare this with the acoustic backscatter anomaly. So this is the anomaly in the acoustic backscatter relative to the time average at each depth. So where we see darker colors means there's more animals at that depth relative to the average. And so you see the diel vertical migration and a fairly strong layer that's quite distinctive that reaches about 300 meters each day. And then what I did upon seeing this, the first thing that clued me in was like day 245 and a half, right? That daytime, there was this distinctive sort of change in the DSL, and at the same time there was a decrease in the surface PAR as reflected by those isolumes. So I was wondering if we could sort of do a better job than what this post-processed ADCP data had in terms of vertical resolution. And the reason that you can do this is because what's optimal binning for an ADCP, which is, for this frequency 8-meter bins, is actually more coarse than what would be sort of optimal if you were just looking at scattering. So an EK instrument, EK80, for example, that many ships are now being equipped with, which is very, very exciting, it typically has a higher vertical resolution because the scale that you can resolve from just acoustic backscatter is finer than what might be optimal for an ADCP. In this case, I worked backwards a little bit, right? So so each of those black dots is the location of an 8-meter bin. And now we can objectively map the shape of the deep scattering layer, the scattering anomaly, which is shown in the dots here, so that we can make a guess as to what the peak of that layer is in between the 8-meter bends. And so that's what's shown with the star, is the peak of the maximum of the deep scattering anomaly over this fairly narrow depth range in between these bends. And so this allowed me to make a sort of higher vertical resolution guess at where the DSL was. And so here's a comparison of the first approach, which is just finding where the maximum is according to the 8-meter binning, and the bottom approach, which is based on objective mapping. The black line shows the extracted depth of the deep scattering layer, and the white line in the background is the isolume depth, and the colors is the backscattering anomaly. So you can already see that you're able to actually resolve and extract some much finer detail out of the objectively mapped data than would have been apparent with the standard ADCP binning. And so here's now those 4 figures all stacked together. We have the surface par, the wire walker measurement, the modeled par subsurface, and then the scattering anomaly with the layer depth in black extracted. And this is just, I just analyzed it between about 7 a.m. and like 7 p.m. or so each day. And then what I did was I wanted to remove, so I was excited about the correlation here, but clearly there's also the migration correlation, the diel migration sort of curve that's imposed on top of it. I wanted to completely remove that so that we can just look at the high frequency component of this behavior. And so for that, I did a running median for the data in both par and surface par and the deep scattering depth layer, and then subtracted that from my signal. So each of the time series here now is just the high pass anomalies with that background signal subtracted. So we see a really impressive correlation on many of these days. Almost every single one of the sort of 25 days that we were out there and I analyzed had a pretty good correlation. Day 242 is one that I put in there that didn't have such a high correlation. And you can see in that case that the variability in the par, which is shown in black now, is sort of varying very quickly and with fairly small amplitude compared to the variations in the depth anomalies, which is shown in red. So I suspect my hypothesis here is that variability in par is either too rapid or maybe is an artifact of something going on the ship that really the animals were not responding to. But with many other of these small variations on the order of 10 minutes, 15 minutes, half hour, there was a pretty remarkable sort of response. And the other thing that was sort of exciting or cool about this data was, I mentioned the importance of the time binning, is that there was a lag. And so you can see it really well on day 232. The par varies, and then sometime later you see the correlation or a similar kind of response to the deep scattering layer. And so we can look at the lagged correlation between these two records and find the maximum. And what that shows is that there's actually a maximum correlation, typically about 5 minutes after the variation in the par. So this is like another kind of piece of the puzzle, another clue that can help us gain some insights into what are the cues for this particular animal layer. And so with this kind of, like, it's so rare to have in situ measurements of animal behavior in conjunction with an environmental parameter like this that is completely independent and yet is so powerfully correlated. And so it's a perfect case for doing a simple model. So the model that I chose, that I sort of landed on, I tried a few different things, including looking at the rate of change of the light, which didn't work. The best model was one that just said, okay, if you're at a particular light level and it's less than what your target, some target isolume is. And what I found for this animal layer is that the target isolume was pretty well described by 0.001 micro-Einsteins per meter squared per second. So if it's less than that, then you want to swim up. And if it's more than that, so it's too bright, then you want to swim down. And if it's within about 10% of your target, then you just stay where you are. And so that model, represented mathematically is shown at the bottom here. Oh, the other thing to mention is that I again, sort of iteratively chose 2 millimeters per second that seemed to do the best job in describing the variability later on. And 2 millimeters per second is well within the swimming sort of capabilities of all of these vertically migrating animals. To put that into a little bit of context, an analysis by Daniele Bianchi found that the average swimming speed during diel vertical migration was about 7 to 10 centimeters per second. All right, so this is now looking at a single day. It's my favorite day. It's the best one, I think, with PAR at the top, and then the modeled PAR subsurface showing the isolumes in white. And then on the bottom, we have our scattering anomaly with our DSL depth in black, and then our model shown in pink. And so you can see that the model actually reproduces a whole bunch of elements. You know, the model is just driven by PAR, but it reproduces a bunch of the elements of the DSL behavior, which is different from just the direct isolume. So it captures the lag, and it also sort of smooths over the corners. It captures the amplitude and a little bit of the kind of the smoothing sort of function that goes into not really responding to either small variations in PAR or really rapid variations. So now we can look at the correlation coefficient for every single one of these days that we analyzed during the cruise. The black is the direct correlation just between the deep scattering anomaly and the PAR anomaly, and again, contains no diel vertical migration. This is just the high frequency component. And the gray shows the sort of small improvement on correlation that you get when you lag it by the optimal lag, typically about 5 minutes. And then the pink is the model. The model does so much better, even for days where the correlation was actually fairly weak. What this is telling me is that they're actually, you know, by applying that model to even days when there wasn't an obvious correlation in PAR, we were still able to get a really good guess at what would be going on with that deep scattering layer. And so that was pretty exciting. And then to quantify that lag time, we see the time series here. It was on average about 4 minutes. And then we can kind of look at or think about what are the, why does this matter? What are some of the bigger implications here? And one of the questions that we had was, what maybe are some of the implications for the distance traveled or the energetics behind doing this high frequency migration, HFM, compared to the diel vertical migration. So on the bottom panel, we have the distance traveled every day by this animal layer, assuming that they're going up into sort of where we can detect them in around the mix layer. down to 300 meters every day. It's about 600-meter distance traveled for the diel component. And then I can integrate the model just for that period of daytime for the high frequency component of the migration, and find that that is on average about 36% of the total distance traveled for diel migration. So about 1/3 of the effort that went into diel vertical migration is now being invested on top of that to do this basically constant swimming response and small adjustment in your vertical depth as a response to clouds. And I wanted to point out that that's actually still conservative, because that estimate doesn't include internal waves. And so this top panel is now an example of the variability in the isopycnals from the wire walker at this site. And we find that the internal wave, the isopycnal excursions, were sort of comparable to some of the scales of excursions from the isolumes. So in order to maintain an isolume depth, which is what it clearly seems that these animals are doing, they have to not only swim in response to the variations in the clouds, but they have to additionally swim in response to displacements that are being imposed on them through internal waves. And so in summary, the migrators that we observed were swimming nearly constantly every day throughout the day. This in itself, I think, is kind of cool, because there are some evidence and some sort of paradigms that have suggested that one of the reasons for doing diel migration might be to sort of slow your metabolism down at depth and kind of rest. And clearly, at least for these animals and this situation, their sort of innate, their evolutionary sort of trait to essentially avoid visual predators here and respond to variability in light is enough to overcome that. So they're swimming all the time. There's a lag, so as the sunlight changes, the layer moves slightly after that. And that can be explained by the fact that it takes you some time to respond. So they're swimming at a rate of about 2 centimeters per second, it looks like. And so that lag can be explained by that time scale. And then there's also a damped response. So it smooths over very rapid light fluctuations. And all of this was really well done with just this simple kind of modeled equation. So the energy that was invested in response to these transient cloud variations is about 1/3 of what is spent on the diel portion of the migration, and that is likely conservative because of internal waves. And so some discussion points I wanted to sort of end with is that one thing that I think really helped us in this system was that we were in a Lagrangian frame of reference. So we weren't constantly moving through fronts and other features that would have led to a kind of, a spatial patchiness or community variation that would have made it more difficult to isolate this behavior. I think it's exciting that these observations were, the primary, most important observations here were just instruments that are commonly available on all research vessels. So there's the potential to look at this with instruments that are already available, perhaps do a meta-analysis of this with archived data. So we just used surface PAR and we used the ADCP for the DSL depth. Options for this open up even more when we can use the EK instruments. And these results support the isolume preferendum idea, the fact that there's a preferred light intensity for a particular species or community, that and that the animals are responding to the absolute light intensity and not the rate of change or other cues. And so thank you so much. I want to thank the NASA OBB program for their support, the export science team, and the crew of the Revell. Take any questions.
Brilliant. Thank you very much, Melissa. That was super cool. Really exciting to see that work coming out. So I'm conscious of the time, so I just want to release anyone who would like to leave now. But I'm really excited about questions, so please do stay on. I have one myself, but I see other people do also have one. So someone raised their hand. Nicholas, please go ahead.
Hi, Melissa. Really interesting work. If you were an organism that was lazy, you'd stay just 50 or 60 meters deeper. So there's an energetic cost to coming back up.
I know, right?
Obviously, it's compensated by whatever they're going to get up. And so did you try to figure out what was it? I mean, you know, looking at particles or changes in particles with depth, what are they gaining from these rapid migrations?
I don't know. We haven't really figured it out, but it's such a good point, right? Like, you know, you might think, well, you just want to be right at the threshold of where your predators can see you. And so maybe that's why you're constantly ducking in and out and trying to maintain this isolume. Whereas the obvious solution to save energy in that case is just to go a little bit deeper, and then you're always below that threshold. So yeah, I think that's a great question. And there wasn't anything really obvious about that, about the horizon of particles or anything at that depth that would suggest that there's like feeding going on or anything like that. So I think it's really open still.
Thanks.
Yeah, but if anyone else in this community has ideas, I think that it's, there was a really good suggestion by Simon Thorold, who was saying that it could be the photophores on the bellies of animals like euphausids, where they'll bioluminesce the right amount to basically self-shade or stay hidden from predators. And so in that case, you can imagine if you did just try to do the solution of going a bit deeper, and then you've got light variations behind you, now there might be a kind of flickering or a variation that would make you susceptible to predators from below. I think that's a great hypothesis.
Nice. Giorgio.
Yeah.
Hi, Melissa. Thank you very much. Really, really interesting. I'm particularly interested because I've worked on a similar topic, but looking at eels migrations. They go down to 700 meters. I'm pretty sure they can detect the clouds from down there. They respond to the full moon or not the full moon. Really exciting. So one reason why eels don't go deeper than a certain depth is that they're sensitive to temperature. Now, of course, they go down to 500-700 meters, so there's a big change in temperature from the surface or from near surface where they go at night. So they don't seem to go below 5 degrees, it seems. But I had a question about the internal waves that you mentioned at the end of the talk. And I was wondering if you've thought about using the residuals between your model and observation to see if you can back estimate the internal waves, which can then validate. Have you tried that?
I would love to. The problem is that even though we were following the wire walker, we were measuring, the ADCP was on the ship, and the internal waves were measured from the wire walker. So they were a little bit separated enough that I don't think there was a perfect sort of phase or matchup between the internal wave field under the ship compared to the wire walker. So we can't back that out. If we had perfectly adjacent measurements, like if we put an ADCP on the wire walker, for example, then I think you could do this. So I'd love to like put an ADCP, maybe some other like optical UVP type measurements on the wire walker, have the buoy showing you surface par, you've got some profiling par on that. I think that would be a great way to look at this.
Interesting. Very, very interesting.
Cool. Jessica, last question.
Hi, Melissa. This is really fascinating. One thing that I was thinking about is that if you're looking at fish, there's often a color difference between a gradient difference between the looking down on the fish versus looking up. And so that difference may actually explain, you know, the preferred isolume that you're looking that the organisms are. But I was kind of wondering, kind of a different question is, do you have a sense of, you know, from different locations, what this preferred isolume might be? Like how, I know that you empirically determined this preference, but, you know,
well,
do you have a sense? Just,
I mean, I'm pretty confident. I'm pretty confident of what the isolume was in this setting. But there were, you know, all kinds of other layers. You know, we were just pulling out the most distinct one. It was very sharp and thin, and likely was comprised of a community that's different from the communities that are going to different depths. And so I think there are lots and lots of layers. And that will, you know, that same community of whatever animal it was that was doing the really sharp, distinct high frequency migration might have a different preference in a different place. So yeah, I don't know, but I think it would also be, you know, it's exciting to think of trying to apply this or look for this in other settings.
Great, thank you.
Brilliant. Thank you so much. Super interesting. And you actually just answered my question as well. So thank you. Okay, so thank you everyone for sticking out a bit longer and for coming in the first place. Please do spread the love. And please do contact the speakers if you have questions. I'm sure they would be happy to hear your suggestions and answer things. So hopefully see you for the next talk and see you then. Bye-bye.